Hi, it's Brett the lab tech here, and I am here to do exercise 4.1 from the Investigating Biology Laboratory Manual. So this is an enzyme experiment that is looking at the action of catecholase, which is an enzyme found in potatoes, and I have some freshly made potato extract right here, and some catechol, which is the substrate for this enzyme. And I'm going to set up the tubes as described in table 4.1 of the lab manual. So the first thing I want to do is label some tubes. Just going to label them as indicated. So we have tube 1, tube 2, and tube 3. Okay. Hopefully you can see those numbers. And now we are going to start adding the contents for the experimental tubes. So I'm going to be taking some pictures throughout the process. So if there's weird pauses in the video, that's what I'm doing. Okay, the first thing we need is some, it says distilled water. Here we only have deionized water, so that's what I'm going to use. And we need 5 milliliters in each of the tubes. Alright, so we have our 5 milliliters of water in each of those tubes. And the next thing to add is catechol. So only tube 1 and 2 are going to get catechol. They're going to get half a milliliter. And we'll add that. Here we've got a 1 milliliter pipette, and I'm going to the half milliliter mark, which is there. All right. We have our catechol, and now we need some more water, deionized water, in tubes 1 and 3. We just need half a milliliter. And here I'm going to sense this maybe a little bit differently. We go up to the 0 mark and carefully dispense half a milliliter. That's tube 1 and tube 3. And this is just so that the final volumes of all these tubes is the same. Okay. Cross-contaminate. Okay, and then the last thing to add is the potato extract. So we are going to add that. And again, half a milliliter into tubes 2 and 3. So exactly a milliliter. Okay. Okay. 2 and tube 3. Okay. I have their potato extract. And now we're going to cover them with, cover each tube with parafilm and invert it a couple of times to mix the reagents really well. The tube 1 again that I'm working on here just has water and catechol. Tube 2 has water and catechol and potato extract. And tube 3 has water and potato extract. All right, so from this you can maybe see some differences. Actually, here I'm going to put these on a different thing to get a little better look at the labels. So the labels are hard to read, but it's tube 1, tube 2, and tube 3. Okay, and those are the results. So record the results in your lab manual and answer the questions.